Good morning, everybody. Today, my team and I would like to present to you a preliminary study on the development of a QCM-based biosensor for EGFR detection in liquid biopsy samples from non-small cell lung cancer patients. This is the outline. First, I'd like to briefly introduce the background of this study. Cancer remains one of the leading causes of death worldwide, with approximately 10 million deaths globally in 2021. In addition, around 1 in 5 people will develop cancer during their lifetime. Among all the cancer types, lung cancer is one of the most serious and deadly diseases. Currently, EGFR mutation detection is commonly performed using tissue biopsy, which involves collecting tumor tissue directly from the patient through invasive procedures such as surgery, which also has several limitations. Repeated tissue sampling for monitoring purposes is not always practical, and in some patients, obtaining sufficient tumor tissue may be difficult. Because of these limitations, liquid biopsy has gained popularity. Liquid biopsy uses blood samples to detect cancer-related biomarkers, such as circulating tumor DNA, instead of collecting tissue directly from the tumor. This method is less invasive, safer, faster, and more convenient for monitoring purposes. In non-small cell lung cancer, or NSCLC, mutations in the EGFR genes are commonly found and are clinically important because they affect the patient's response to targeted anti-EGFR therapies. Important EGFR mutations genes include exon 18, 19, 20, and 21. However, conventional EGFR mutation tests are relatively expensive, costing around 6,000 to 8,000 baht per test. Therefore, there is a need for a rapid, sensitive, and more cost-effective detection platform. To address this problem, our study focused on developing a piezoelectric biosensor based on QCM technology. The principle of the QCM biosensorship is based on the resonance frequency changes on the quartz crystal surface. When biomolecules bind to the surface, the mass increases, therefore the resonance frequency decreases. This frequency shift can then be used to detect the DNA hybridization events. The objectives of this study were to first, investigate the EGFR gene mutation using QCM biosensor platforms. Second, to optimize important experimental conditions such as probe immobilization, probe target hybridization, and plasma dilution ratios. And third, to evaluate the plasma samples by correlating PCR-CT values with the resonance frequency shifts obtained from the QCM biosensor. Our hypothesis was that EGFR mutations could be effectively detected using this biosensor platform. Temperature, incubation time, and plasma ratio would significantly affect the detection performance. We also hypothesized that exon 18 and exon 19 mutations would selectively hybridize with their complementary probes on the QCM surface.
For experimental procedure, first, the QCM chip will clean in pure solution, then ultrasonic cleaning in acetone and ethanol. Lastly, in deionized water. After that, single-strand DNA probe were immobilized onto the QCM chip surface. The resonance frequency shifts were measured before and after probe attachment, target hybridization, and plasma application before plot graph. Several parameters were optimized in this study. For probe immobilization, incubation temperature ranged from 25 to 60 degrees Celsius, and incubation times range from 10 minutes to overnight. For probe target hybridization, incubation temperature ranged from 25 to 65 degrees Celsius, and incubation time ranged from 5 to 60 minutes. We also optimized plasma dilution ratios and compare exon 18 and exon 19 plasma detection performance. For probe immobilization optimization, we evaluate resonant frequency shifts across varying incubation times at room temperature and at 60 degrees Celsius. By comparing these two experimental conditions, we observed that both graphs consistently indicated the same optimal conditions, which was at 60 degrees Celsius for 120 minutes. This have confirmed that the selected temperature and incubation time were provided the most effective probe immobilization on the QCM electrode surface. In total, 71 independent sensor trials were performed to evaluate probe binding performance under the optimal condition. The experiment achieved a 66% success rate in EGFR single-strand DNA probe attachment, indicating effective and reproducible probe immobilization. Next, we optimized probe target hybridization conditions. The results demonstrate that 30 degrees Celsius and 30 minutes What's the optimal hybridization temperature and time for achieving maximum probe target binding? These conditions generate the most reliable resonant frequency response. We then optimize plasma dilution conditions. The results show that a plasma dilution ratio of 1 to 200 BTE buffer produced the highest resonant frequency shifts. Higher plasma concentrations cause non-specific binding and protein interference, while lower concentrations weaken detection clarity. Figure 7 shows the comparison of resonant frequency shifts among the experimental group after plasma detection. The mutation-positive plasma samples produced larger frequency shifts compared with the negative controls, indicating successful hybridization between the EGFR targets and their complementary probes on the QCM surface. These results demonstrate that the QCM biosensor can successfully detect EGFR mutations from liquid biopsy plasma samples. Finally, we evaluate plasma detection performance for exon 18 and exon 19 mutations. The results show that plasma sample 1 and 2 follow an inverse relationship between PCR CT values and QCM frequency shifts. Lower CT values corresponded to larger frequency shifts, supporting the semi-quantitative capability of the QCM biosensor platform. In addition, both exon 18 and exon 19 probes demonstrated selective detection of their respective targets, with only minor cross-recognition observed between mutations. In conclusion, the optimal probe immobilization condition was 60 degrees Celsius for 120 minutes with 66% success rate, while the optimal hybridization condition was 30 degrees Celsius for 30 minutes. A plasma dilution ratio of 1 to 200 provided the best resonant frequency response across repeated measurements. Importantly, the inverse relationship between PCR CT values and QCM resonant frequency shifts confirmed the semi-quantitative detection capability of the system. Overall, the QCM biosensor successfully differentiated EGFR variants and demonstrated strong potential as a rapid and cost-effective tool for liquid biopsy application in lung cancer detection.